Insect reproductive system. Most insects reproduce oviparously, i.e. by laying eggs. The eggs are produced by the female in a pair of ovaries. Sperm, produced by the male in one testicle or more commonly two, is transmitted to the female during mating by means of external genitalia. The sperm is stored within the female in one or more spermathecae. At the time of fertilization, the eggs travel along oviducts to be fertilized by the sperm and are then expelled from the body, laid, in most cases via an ovipositor. Internal. Female. The female genitalia of Lepidoptera. Female insects are able to make eggs, receive and store sperm, manipulate sperm from different males, and lay. Female insects are able to make eggs, receive and store sperm, manipulate sperm from different males, and lay eggs. Their reproductive systems are made up of a pair of ovaries, accessory glands, one or more spermathecae, and ducts connecting these parts. The ovaries make eggs and accessory glands produce the substances to help package and lay the eggs. Spermathecae store sperm for varying periods of time and, along with portions of the oviducts, can control sperm use. The ducts and spermathecae are lined with a cuticle. 1-880. The ovaries are made up of a number of egg tubes, called ovarioles, which vary in size and number by species. The number of eggs that the insect is able to make varies according to the number of ovarioles, with the rate at which eggs develop being also influenced by ovariole design. In meroistic ovaries, the eggs to be divide repeatedly and most of the daughter cells become helper cells for a single oocyte in the cluster. In panoistic ovaries, each egg to be produced by stem germ cells develops into an oocyte. There are no helper cells from the germ line. The ovaries are made up of a number of egg tubes, called ovarioles, which vary in size and number by species. The number of eggs that the insect is able to make varies according to the number of ovarioles, with the rate at which eggs develop being also influenced by ovariole design. In meroistic ovaries, the eggs to be divide repeatedly and most of the daughter cells become helper cells for a single oocyte in the cluster. In panoistic ovaries, each egg to be produced by stem germ cells develops into an oocyte. There are no helper cells from the germ line. Production of eggs by panoistic ovaries tends to be slower than that by meroistic ovaries. 1-880. Accessory glands are glandular parts of the oviducts produce a variety of substances for sperm maintenance, transport, and fertilization, as well as for protection of eggs. They can produce glue and protective substances for coating eggs or tough coverings for a batch of eggs called oothecae. Spermathecae are tubes or sacs in which sperm can be stored between the time of mating and the time an egg is fertilized. Paternity testing of insects has revealed that some, and probably many, female insects use the spermatheca and various ducts to control or bias sperm used in favor of some males over others. 1-880. Male. Male genitalia of Lepidoptera. The main component of the male reproductive system is the testicle, suspended in the body cavity by tracheae and the fat body. The more primitive apterygote insects have a single testis, and in some lepidopterans the two maturing testes are secondarily fused into one structure during the later stages of larval development, although the ducts leading from them remain separate. However, most male insects have a pair of testes, inside of which are sperm tubes or follicles that are enclosed within a membranous sac. The follicles connect to the vas efferens by the vas deferens, and the two tubular vasa deferentia connect to a median ejaculatory duct that leads to the outside. A portion of the vas deferens is often enlarged to form the seminal vesicle, which stores the sperm before they are discharged into the female. The seminal vesicles have glandular linings that secrete nutrients for nourishment and maintenance of the sperm. The ejaculatory duct is derived from an invagination of the epidermal cells during development and, as a result, has a cuticular lining. The terminal portion of the ejaculatory duct may be sclerotized to form the intermittent organ, the aedegus. The remainder of the male reproductive system is derived from embryonic mesoderm, except for the germ cells, or spermatogonia, which descend from the primordial pole cells very early during embryogenesis. 1-885. External. Variety of male structures in Phlebotominae, Diptera, Psychotidae. The ground plan of the abdomen of an adult insect typically consists of 11 to 12 segments and is less strongly sclerotized than the head or thorax. Each segment of the abdomen is represented by a sclerotized tergum, sternum, and perhaps a pleurite. Terga are separated from each other and from the adjacent sterna or pleura by a membrane. Spiracles are located in the pleural area. Variation of this ground plan includes the fusion of turga or turga and sterna to form continuous dorsal or ventral shields or a conical tube. Some insects bear a sclerite in the pleural area called a laterotergite. Ventral sclerites are sometimes called laterosternites. During the embryonic stage of many insects and the postembryonic stage of primitive insects, 11 abdominal segments are present. In modern insects there is a tendency toward reduction in the number of the abdominal segments, but the primitive number of 11 is maintained during embryogenesis. Variation in abdominal segment number is considerable. If the apterogota are considered to be indicative of the ground plan for PT or egoats, confusion reigns. Adult Protura have 12 segments, Collembola have 6. The Orthopteran family Acrididae has 11 segments, and a fossil specimen of Zoroptera has a 10-segmented abdomen. 1. Generally, the first 7 abdominal segments of adults, the progenital segments, are similar in structure and lack appendages. However, apterogoats, bristletails and silverfish, and many immature aquatic insects have abdominal appendages. Apterogoats possess a pair of styles, rudimentary appendages that are serially homologous with the distal part of the thoracic legs. And, messily, 1 or 2 pairs of protrusible or exertile vesicles on at least some abdominal segments. These vesicles are derived from the coxal and trochantral indites, inner annulated lobes, of the ancestral abdominal appendages. Aquatic larvae and nymphs may have gills laterally on some to most abdominal segments. 249 of the rest of the abdominal segments consist of the reproductive and anal parts. The anal genital part of the abdomen, known as the terminalia, consists generally of segments 8 or 9 to the abdominal apex. Segments 8 and 9 bear the genitalia. Segment 10 is visible as a complete segment in many lower insects but always lacks appendages, and the small segment 11 is represented by a dorsal epiproct and pair of ventral paraprocts derived from the sternum. A pair of appendages, the cerci, articulates laterally on segment 11. Typically these are annulated and filamentous but have been modified, e.g. the forceps of earwigs, or reduced in different insect orders. An annulated caudal filament, the median appendix dorsalis, arises from the tip of the epiproct in apterogoats, most mayflies, Ephemeroptera, and a few fossil insects. A similar structure in nymphal stoneflies, Plecoptera, is of uncertain homology. These terminal abdominal segments have excretory and sensory functions in all insects, but in adults there is an additional reproductive function. 249. Genitalia. The organs concerned specifically with mating and the deposition of eggs are known collectively as the external genitalia, although they may be largely internal. Insect genitalia, especially male genitalia, is often directionally asymmetrical, and this trait has evolved multiple times in various orders. 3. The components of the external genitalia of insects are very diverse in form and often have considerable taxonomic value, particularly among species that appear structurally similar in other respects. The male external genitalia have been used widely to aid in distinguishing species, whereas the female external genitalia may be simpler and less varied. The terminalia of adult female insects include internal structures for receiving the male copulatory organ and his spermatozoa, and external structures used for oviposition, egg laying. Most female insects have an egg-laying tube, or ovipositor. It is absent in termites, parasitic lice, many Plecoptera, and most Ephemeroptera. Ovipositors take 2 forms. 1. True, or appendicular, formed from appendages of abdominal segments 8 and 9. 2. Substitutional, composed of extensible posterior abdominal segments. Meiosis. Meiosis is a fundamental aspect of insect reproductive systems, and likely has an adaptive function similar to that of meiosis in eukaryotes generally. Studies of meiosis in the insect Drosophila melanogaster at the molecular level have provided insight into the adaptive function of meiosis. Mutants of D. melanogaster that are defective in genes MAY41 and MAY9 that encode products employed in meiosis were found to display decreased meiotic recombination and also to have increased sensitivity to the DNA-damaging agents X-rays, UV, methylmethane sulfonate and nitrogen mustard. 4. These results link meiotic recombination to DNA repair and suggest that a function of recombination during meiosis in D. melanogaster is repair of damages in germline DNA. Evolution of insects. The most recent understanding of the evolution of insects is based on studies of the following branches of science: molecular biology, insect morphology, paleontology, insect taxonomy, evolution, embryology, bioinformatics and scientific computing. It is estimated that the class of insects originated on Earth about 480 million years ago, in the Ordovician, at about the same time terrestrial plants appeared. 1. Insects are thought to have evolved from a group of crustaceans. 2. The first insects were landbound, but about 400 million years ago in the Devonian period one lineage of insects evolved flight, the first animals to do so. 1. The oldest insect fossil has been proposed to be Rhyniagnatha hursti, estimated to be 400 million years old, but the insect identity of the fossil has been contested. 3. Global climate conditions changed several times during the history of Earth, and along with it the diversity of insects. The pterygotes, winged insects, underwent a major radiation in the Carboniferous, 358 to 299 million years ago, while the Endopterygota, insects that go through different life stages with metamorphosis, underwent another major radiation in the Permian, 299 to 252 million years ago. Evolution has produced astonishing variety of appendages in insects, such as these antennae. Most extant orders of insects developed during the Permian period. Many of the early groups became extinct during the mass extinction at the Permo-Triassic boundary, the largest extinction event in the history of the Earth, around 252 million years ago. 4. The survivors of this event evolved in the Triassic, 252 to 201 million years ago, to what are essentially the modern insect orders that persist to this day. Most modern insect families appeared in the Jurassic, 201 to 145 million years ago. In an important example of coevolution, a number of highly successful insect groups, especially the Hymenoptera wasps, bees and ants, and Lepidoptera butterflies, as well as many types of Diptera flies and Coleoptera beetles evolved in conjunction with flowering plants during the Cretaceous, 145 to 66 million years ago. 5. 6. Many modern insect genera developed during the Cenozoic that began about 66 million years ago. Insects from this period onwards frequently became preserved in amber, often in perfect condition. Such specimens are easily compared with modern species, and most of them are members of extant genera. Fossils. Preservation. Fossil net in amber, a well-preserved insect fossil. Due to their external skeleton, the fossil history of insects is not entirely dependent on Lagerstätte-type preservation as for many soft-bodied organisms. However, with their small size and light build, insects have not left a particularly robust fossil record. Other than insects preserved in amber, most finds are terrestrial or near-terrestrial sources and only preserved under very special conditions such as at the edge of freshwater lakes. While some 1/3 of known non-insect species are extinct fossils, due to the paucity of their fossil record, only 1/100th of known insects are extinct fossils. 7. Insect fossils are often three-dimensional preservations of the original fossil. Loose wings are a common type of fossil as the wings do not readily decay or digest, and are often left behind by predators. Fossilization will often preserve their outer appearance, contrary to vertebrate fossils whom are mostly preserved just as bony remains or inorganic casts thereof. Due to their size, vertebrate fossils with the external aspect similarly preserved are rare, and most known cases are subfossils. 8. Fossils of insects, when preserved, are often preserved as three-dimensional, permineralized, and charcoalified replicas, and as inclusions in amber and even within some minerals. Sometimes even their color and patterning is still discernible. 9. Preservation in amber is, however, limited since copious resin production by trees only evolved in the Mesozoic. 10. 11. There is also abundant fossil evidence for the behavior of extinct insects, including feeding damage on fossil vegetation and in wood, fecal pellets, and nests in fossil soils. Such preservation is rare in vertebrates, and is mostly confined to footprints and coprolites. 1242. Freshwater and marine insect fossils. The common denominator among most deposits of fossil insects and terrestrial plants is the lake environment. Those insects that became preserved were either living in the fossil lake, autochthonous, or carried into it from surrounding habitats by winds, stream currents, or their own flight, allochthonous. Drowning and dying insects not eaten by fish and other predators settle to the bottom, where they may be preserved in the lake's sediments, called lacustrine, under appropriate conditions. Even amber, or fossil resin from trees, requires a watery environment that is lacustrine or brackish in order to be preserved. Without protection in anoxic sediments, amber would gradually disintegrate. It is never found buried in fossil soils. Various factors contribute greatly to what kinds of insects become preserved and how well, if indeed at all, including lake depth, temperature, and alkalinity, type of sediments, whether the lake was surrounded by forest or vast and featureless salt pans, and if it was choked in anoxia or highly oxygenated. There are some major exceptions to the lacustrine theme of fossil insects, the most famous being the late Jurassic limestones from Zonhofen and Eichstätt, Germany, which are marine. These deposits are famous for pterosaurs and the bird-like Archaeopteryx. The limestones were formed by a very fine mud of calcite that settled within stagnant, hypersaline bays isolated from inland seas. Most organisms in these limestones, including rare insects, were preserved intact, sometimes with feathers and outlines of soft wing membranes, indicating that there was very little decay. The insects, however, are like casts or molds, having relief but little detail. In some cases iron oxides precipitated around wing veins, revealing better detail. 1242. Compressions, impressions and mineralization. There are many different ways insects can be fossilized and preserved, including compressions and impressions, concretions, mineral replication, charcoalified fusainized remains, and their trace remains. Compressions and impressions are the most extensive types of insect fossils, occurring in rocks from the Carboniferous to the Holocene. Impressions are like a cast or mold of a fossil insect, showing its form and even some relief, like pleating in the wings, but usually little or no color from the cuticle. Compressions preserve remains of the cuticle, so color distinguishes structure. In exceptional situations, microscopic features such as microtrichia on sclerites and wing membranes are even visible, but preservation of this scale also requires a matrix of exceptionally fine grain, such as in micritic muds and volcanic tuffs. Because arthropod sclerites are held together by membranes, which readily decompose, many fossil arthropods are known only by isolated sclerites. Far more desirable are complete fossils. Concretions are stones with a fossil at the core whose chemical composition differs from that of the surrounding matrix, usually formed as a result of mineral precipitation from decaying organisms. The most significant deposit consists of various localities of the late Carboniferous Francis Creek Shale of the Carbondale Formation at Mazon Creek, Illinois, which are composed of shales and coal seams yielding oblong concretions. Within most concretions is a mold of an animal and sometimes a plant that is usually marine in origin. When an insect is partly or wholly replaced by minerals, usually completely articulated and with three-dimensional fidelity, is called mineral replication. 12. This is also called petrifaction, as in petrified wood. Insects preserved this way are often, but not always, preserved as concretions, or within nodules of minerals that formed around the insect as its nucleus. Such deposits generally form where the sediments and water are laden with minerals, and where there is also quick mineralization of the carcass by coats of bacteria. Evolutionary history. The insect fossil record extends back some 400 million years to the Lower Devonian, while the pterygotes, winged insects, underwent a major radiation in the Carboniferous. The Endopterygota underwent another major radiation in the Permian. Survivors of the mass extinction at the PT boundary evolved in the Triassic to what are essentially the modern insecta orders that persist to modern times. Most modern insect families appeared in the Jurassic, and further diversification probably in genera occurred in the Cretaceous. By the Tertiary, there existed many of what are still modern genera. Hence, most insects in amber are, indeed, members of extant genera. Insects diversified in only about 100 million years into essentially modern forms. 7. Insect evolution is characterized by rapid adaptation due to selective pressures exerted by the environment and furthered by high fecundity. It appears that rapid radiations and the appearance of new species, a process that continues to this day, result in insects filling all available environmental niches. The evolution of insects is closely related to the evolution of flowering plants. Insect adaptations include feeding on flowers and related structures, with some 20% of extant insects depending on flowers, nectar or pollen for their food source. This symbiotic relationship is even more paramount in evolution considering that more than 2/3 of flowering plants are insect-pollinated. 13. Insects, particularly mosquitoes and flies, are also vectors of many pathogens that may even have been responsible for the decimation or extinction of some mammalian species. 14. Before the Devonian. Molecular analysis by Gunt and Miles 2002 suggests that the hexapods diverged from their sister group, the Anastraca fairy shrimps, at around the start of the Silurian period 440 million years ago, coinciding with the appearance of vascular plants on land. 15. Massoff et al. Suggest that insects could have appeared much earlier, in the early Ordovician or even Cambrian. According to this version, the early radiation of insects occurred no later than 479 million years ago in marine or coastal environments. However, the authors emphasize that due to the lack of insect fossils of the Cambrian to the Silurian, this version remains highly controversial. 16. Devonian. Reconstruction. Strudiella devonica is a possible hexapod. The Devonian, 419 to 359 million years ago, was a relatively warm period, and probably lacked any glaciers. The details of early insect fossil records are not well understood. The fossils that were considered as Devonian insects, such as Rhyniagnatha hursti, 17, or Strudiella devonica, 18, were later reconsidered that their affinities as insects are insufficient. 319 But based on phylogenetic study, the first insects probably appeared earlier, in the Silurian period, 16, from stem group crustaceans like Tanasiostocaran, 20, that had lost the second antenna. The first winged insect likely evolved in the Devonian given the appearance of large numbers of insects with wings in the Carboniferous. 9. Carboniferous. Mazotharos, a Carboniferous member of the now extinct order Paleodictyoptera. The Carboniferous. 359 to 299 million years ago, is famous for its wet, warm climates and extensive swamps of mosses, ferns, horsetails, and calamites. 21. Glaciations in Gondwana, triggered by Gondwana's southward movement, continued into the Permian, and because of the lack of clear markers and breaks, the deposits of this glacial period are often referred to as Permo-Carboniferous in age. The cooling and drying of the climate led to the Carboniferous rainforest collapse (CRC). Tropical rainforests fragmented and then were eventually devastated by climate change. 22. Remains of insects are scattered throughout the coal deposits, particularly of wings from Stemdictyopterans Blattoptera. 23. Two deposits in particular are from Mazon Creek, Illinois and Commentry, France. 24. The earliest winged insects are from this time period Pterygota, including the aforementioned Blattoptera, Colonerodia, primitive stem group Ephemeropterans, Orthoptera, Paleodictyopteroidea. 21. 399 In 1940, in Noble County, Oklahoma, a fossil of Meganeuropsis americana represented the largest complete insect wing ever found. 25. Juvenile insects are also known from the Carboniferous period. 26. Very early Blattopterans had a large, discoid pronotum and coriaceous forewings with a distinct cup vein, a unbranched wing vein, lying near the claval fold and reaching the wing posterior margin. These were not true cockroaches, as they had an ovipositor, although through the Carboniferous, the ovipositor started to diminish. The orders Colonerodia and Myomoptera are known, with Orthoptera and Blatodia to be among the earliest Neoptera, developing from the Upper Carboniferous to the Permian. These insects had wings with similar form and structure, small anal lobes. 21. 399 species of Orthoptera, or grasshoppers and related kin, is an ancient order that still exists till today, extending from this time period. From which time even the distinctive synapomorphy of saltatorial, or adaptive for jumping, hind legs is preserved.